Your body is accumulating zombie cells, and they are quietly destroying your organs. That is not a metaphor. Scientists actually call them zombie cells, cells that should have died but did not, and now sit inside your tissues actively poisoning the healthy cells around them. The formal term is senescent cells, and the research emerging around them and around the compounds that can selectively clear them represents one of the most genuinely exciting frontiers in longevity science right now. On Monday, we covered chronic inflammation and the 7 compounds proven to reduce it. Today, we are going deeper because senescent cells are one of the primary drivers of that very inflammation. These 2 topics are directly connected at the biological level, and by the end of this video, you will understand exactly how. Let us start with biology. To understand why senescent cells matter, you first need to understand what normally happens when a cell reaches the end of its useful life. Every cell in your body has a built-in life cycle. It divides, performs its function, and eventually reaches a point where it can no longer divide safely. At that point, the cell faces 2 options. The first option is apoptosis, programmed cell death. The cell self-destructs in a controlled, clean manner. Its components are recycled by the body, and the tissue renews. This is healthy and normal. The second option is cellular senescence. Instead of dying, the cell stops dividing but remains metabolically active inside your tissue. It does not perform its original function. It does not recycle itself. It simply stays permanently and begins releasing a cocktail of inflammatory molecules that damage surrounding tissue. This cocktail has a name: the senescence-associated secretory phenotype, or SASP. It includes pro-inflammatory cytokines, the same IL-6 and TNF-alpha we discussed on Monday, along with proteases that degrade surrounding tissue structure and growth factors that can promote abnormal behavior in neighboring cells. Think of a senescent cell as a cell that refused to follow its programmed exit and is now actively disrupting the neighborhood. In young tissue, senescent cells appear transiently. They form as part of wound healing and tumor suppression, then are cleared efficiently by the immune system within days or weeks. The problem is that with age, 2 things happen simultaneously. Senescent cells accumulate faster, and the immune system becomes less efficient at clearing them. The result is a progressive buildup of SASP-secreting cells across multiple organs. We now know what senescent cells are and what they do. The next question is, does this actually matter for aging in practice? The research answer is unambiguous. The scientific case connecting senescent cell accumulation to aging has been building since 2011. Let me walk you through the landmark studies. The foundational experiment was published in Nature in 2011 by Darren Baker and Jan van Dersen at the Mayo Clinic. They engineered mice to allow selective clearance of senescent cells and found that removing them delayed the onset of cataracts, muscle wasting, and fat tissue dysfunction. The mice lived measurably healthier for longer. In 2016, the same Mayo Clinic team published a follow-up in Nature showing that clearing senescent cells from already aged mice extended their remaining lifespan by 25 to 35% and improved physical, heart, and kidney function. These were not young mice being kept healthy. These were old mice being functionally rejuvenated. A 2018 paper in Nature Medicine from Mayo Clinic and Scripps Research Institute was the first human translation. They studied patients with idiopathic pulmonary fibrosis, a fatal lung disease associated with high senescent cell burden, and found that a combination of dasatinib and quercetin reduced circulating senescent cell markers and improved physical function. Small trial, but proof of concept in living human beings. The scientific consensus is now clear. Senescent cells accumulate with age. Their SASP drives systemic inflammation and tissue dysfunction, and clearing them has meaningful biological effects. The question being actively studied is how to do this safely and effectively at scale in humans. Before we move into the senolytics, the compounds being studied to actually clear these cells, one quick note. If this depth of cellular science is what you have been looking for, this channel publishes exactly this, twice every week, Monday and Friday. Peer-reviewed longevity science explained properly. Not wellness content, not recycled supplement marketing. The actual research. Subscribing takes 1 second and ensures you do not miss what is coming next. So we have established what senescent cells are, why they accumulate, and what decades of research shows about their role in aging. The logical next question is, can we actually do something about them? That is exactly what the field of senolytics is trying to answer. Senolytics are compounds, pharmaceutical or natural, that selectively induce death in senescent cells while leaving healthy cells largely unaffected. Let us go through the 3 natural compounds with the strongest research behind them for you, starting with the most studied. The first senolytic is Fisetin, a flavonoid found naturally in strawberries, apples, and persimmons, and currently the most studied natural senolytic compound in the research literature. A 2018 study published in eBiomedicine by researchers at the Mayo Clinic tested Fisetin against 10 other flavonoids and found it was the most potent for senolytic activity, meaning it most selectively induced death in senescent cells while leaving healthy cells largely unaffected. In aged mice, Fisetin administration restored tissue homeostasis, reduced inflammatory markers, and extended both median and maximum lifespan. These are striking results for a compound found in everyday food. Human clinical trials are currently underway. A trial at the University of Minnesota, registered as NCT03675724, is investigating Fisetin's effects in older adults. We do not yet have published large-scale human RCT results, and I want to be transparent about that. The animal data is compelling. The human data is still being gathered. The doses referenced in the research literature range from 100 to 500 milligrams, sometimes used in an intermittent pulse protocol rather than daily. The reasoning being that senolytic activity may be better achieved in concentrated bursts. Fisetin has the strongest natural senolytic evidence base at this point. The second compound is one you have already heard this week, and the fact that it appears here again tells us something important about how these biological pathways overlap. The second senolytic is quercetin. And yes, this is the same quercetin we covered on Monday as an anti-inflammatory compound. Its reappearance here is not a coincidence. It reflects something genuinely important. Certain compounds operate across multiple biological mechanisms simultaneously, which is part of what makes them so interesting to longevity researchers. Quercetin's senolytic mechanism is distinct from its anti-inflammatory action. As a senolytic, it inhibits anti-apoptotic proteins, specifically BCL2 family proteins, that senescent cells overexpress to avoid dying. By removing this molecular survival shield, quercetin pushes senescent cells back toward programmed death. The Mayo Clinic and Scripps human trial I referenced earlier used quercetin at 250 milligrams combined with the pharmaceutical dasatinib. It is important to note that dasatinib is a prescription cancer drug with significant side effects, not something to self-administer. But the quercetin component of that combination is what makes it directly relevant here. Researchers are now studying quercetin independently, without the pharmaceutical, at doses of 500 to 1,000 milligrams to assess its standalone senolytic activity in humans. So we have Fisetin, the most potent natural senolytic in the current research, and quercetin, which adds a second mechanism and an anti-inflammatory effect simultaneously. The third compound is less well-known, but worth understanding because it illustrates how broad the natural senolytic landscape actually is. The third compound is Piperlongumine, extracted from long pepper, known as Piperlongum. This is not a supplement you will find in every health store, and I want to be clear that the human research base is thinner here than for Fisetin or quercetin. A 2017 study published in Aging Cell found Piperlongumine induced senescent cell death across multiple cell types while maintaining specificity for senescent versus normal cells. It is included here not as a primary recommendation, but because it demonstrates the breadth of naturally occurring compounds now being investigated in this space. As the field matures, Piperlongumine may become more prominent. Before we move to practical takeaways, I want to briefly cover one pharmaceutical agent, not because it is something you would take, but because understanding how it works scientifically strengthens the case for the natural compounds we have just discussed. That compound is called Navitoclax. And unlike everything we have covered today, it is not a supplement. It is a prescription pharmaceutical drug currently used in cancer treatment. I am not suggesting you take it. What I am suggesting is that you understand what it does, because it directly explains why quercetin works the way it does at the cellular level. Navitoclax inhibits BCL2 family proteins, the same anti-apoptotic proteins that quercetin also targets. When BCL2 is inhibited, senescent cells lose their survival advantage and undergo apoptosis. The reason I include it here is purely mechanistic. Navitoclax demonstrates that BCL2 inhibition is a proven, clinically validated senolytic mechanism in human biology. Quercetin operates through a partially overlapping version of that same pathway. This is precisely why researchers take quercetin seriously as a natural senolytic candidate. The underlying mechanism is not speculative, it is validated at the pharmaceutical level. The active question in current trials is whether quercetin achieves sufficient potency and bioavailability at supplement doses to produce a meaningful effect. That data is coming. We have now covered 4 senolytics, from the most studied natural compound to pharmaceutical validation of the mechanism. Before practical recommendations, I want to give you an honest summary of where the science actually stands, because the marketing in this space has run significantly ahead of the clinical evidence. What we know with reasonable confidence: senescent cells accumulate with age and contribute to systemic inflammation and tissue dysfunction. Clearing them in animal models produces measurable improvements in health span. Several natural compounds, particularly Fisetin and Quercetin, have demonstrated senolytic activity in cell culture and animal models with plausible, validated mechanisms of action. What we do not yet know: whether oral supplementation at achievable doses produces meaningful senolytic effects inside a living human body. Bioavailability is a significant variable. The doses required may differ substantially from standard supplement doses, and the long-term safety of periodically inducing apoptosis across cell populations throughout the body has not been rigorously studied. This is an area to watch closely over the next 3 to 5 years as human trial data matures. The scientific rationale is solid, the animal data is compelling, the human data is early but directionally consistent, and the natural compounds involved have reasonable safety profiles at moderate doses. As always, this is not medical advice. If you are on prescription medications or have any underlying health conditions, particularly cancer or immune disorders, discuss supplementation in this area with your physician first. With that honest framing in place, let me give you the practical takeaways, what you can actually do with this information right now. First, the dietary angle is real and underappreciated. Fisetin is found in meaningful concentrations in strawberries, roughly 160 micrograms per gram, and in smaller amounts in apples, grapes, onions, and cucumbers. This is not a substitute for therapeutic doses, but it is a genuinely evidence-based reason to prioritize these specific foods for anyone focused on longevity. Second, if you are considering Fisetin supplementation, the doses referenced in the research literature range from 100 to 500 milligrams. Some researchers advocate for an intermittent pulse protocol, 2 to 3 consecutive days per month at higher doses, rather than daily low-dose supplementation. This remains experimental, but the reasoning is mechanistically sound. Third, the quercetin and curcumin combination from Monday Now has a dual justification: anti-inflammatory activity through NF-κB inhibition, and potential senolytic activity through BCL2 pathway modulation. These are complementary mechanisms addressing overlapping biology, which is why researchers studying both inflammation and senolytics keep arriving at the same short list of compounds. I have reviewed Fisetin and Quercetin products against published purity and bioavailability data from independent testing. The links are in the description below. As always, only products that clear the research criteria make it there. So where does this leave us? Senescent cells represent one of the most tangible biological targets in longevity science today. Something measurable, mechanistically understood, and increasingly actionable. The science is moving quickly, and this channel will cover the human trial results as they arrive. Next week, we move to the brain. We will be looking at the nootropic stack that actually has peer-reviewed evidence behind it, covering lion's mane, bacopa, phosphatidylserine, and magnesium L-threonate, and why the gut-brain inflammation axis we have been building across this week connects directly to cognitive aging. That is a video worth setting a reminder for. If you haven't subscribed yet, this is the moment. Here at the Nutraceutical Scientist, we translate the latest research on longevity, healthy aging, and evidence-based nutraceuticals into practical advice you can use every day. Subscribe, and new episodes arrive every Monday and Friday. Until we meet again this Monday, stay healthy, stay curious, and remember, every healthy choice you make today is an investment in the life you'll live tomorrow.